Hello everyone. So today we'll be discussing an overview of radiobiology. And to begin with, I'm sorry for this long video. I try to keep it as short as possible. Radiobiology is huge. So let's get on with it. The origins of radiobiology's contribution to radiotherapy can actually be traced back to these two lectures. One by Dr. Henry S. Kaplan of Stanford in 1970, and the other by Dr. Herman D. Soot in 1983. He was from Harvard. They both actually discussed how radiobiology actually affects and has led to the development of radiotherapy as such. So, radiotherapy initially was basically based on empiricism. Empiricism means what has worked in the past. What has worked has worked. We'll keep on doing it. So, we basically would rely on, accumulated clinical experience. And there are very strong signs of empiricism in our literature. We have case reports, we have retrospective studies, we have single institutional clinical series, we have a lot of retrospective data, especially regarding radiation volumes, the dose, the treatment techniques. So, the initial development of radiation oncology has been really based on empiricism. We were just doing what we were doing, and if it was working, we were doing it more. Now we have shifted towards research based radiation oncology because now we are understanding the genome, we are understanding the messengers, we are understanding how molecular and targeted therapy works, how the tumor biology is, and how we can get the best for our patient out of it. So, a lot of translational research has been done and is being promoted, and it's very important to actively participate it as as a radiation oncologist. The gold standard that we have is a phase 3 retrospective randomized control trial. So, coming to what are the effects of a radiation on a cell? The first and foremost effect is DNA damage. DNA damage in itself can lead to three things. It can lead to mitotic cell death or apoptosis. Your cell can survive, it can repair itself and come back to the normal status, or it can misrepair, lead to some sort of mutation. This mutation might express itself or not, it might lead to carcinogenesis or not. Basically, we are aiming for the first two things. We are aiming for a cell death for the tumor, and we are aiming for survival and a normal status for the normal tissues. So, this whole physical interaction of the radiation with the molecular infrastructure of the cell takes place in about 10 to the power minus 18 to 10 to the power minus 3 seconds, very quickly. What happens is there's absorption of the photon, there's destabilization of the target molecule, which is usually the DNA. There are molecular breaks, there's a release of energetic electrons, attenuated photons, and there's interaction with other cellular molecules, which leads to a kind of a chain reaction, leading to the birth of short-lived ions and chemically unstable free radicals. So the free radicals that come up are hydroxyl, hydrated electrons, hydrogen atoms, hydrogen peroxide, and it's almost instantaneous. You see 10 to the power minus 18 to 10 to the power 3 seconds. And then these free radicals interact with other neighboring molecules and they produce chemically stable lesions. So there is the production by radiolysis of water, and for the subsequent reactions, there's a production of a lot of free radicals which lead to this DNA damage, and this DNA damage is fixed in the presence of oxygen. So free radical scavengers and oxygen is very important to fix this damage. Now all the damage has been done. What's the cascade now? So this has happened in second. Now after minutes of eradication, there is protein kinase C, and tyrosine kinase mediated pathways which are stimulated. These are basically for the radiation damage repairs. Several stress genes are induced, other growth factors, cytokines are produced. Then there is modulation of the mammalian cells, there is production of tumor necrosis factor, fibroblast growth factor, transforming growth factor, tissue plasminogen activator. And if all these factors remain constant, the permanent damage is linear with the dose. Now, we talked about that there is a target. Experiments have shown that the nucleus requires less dose of irradiation as compared to the cytoplasm. So we have decided that nucleus is the first target, the second being the cell membrane, and the third being the mitochondrial membrane. The general consensus among all people is that the target molecules are there in the nucleus only, and basically it's the DNA. Now, in many cells, this radiation-induced lethality is not instantaneous, that you irradiate and the cell will die. It doesn't work that way. The cell will continue to function. The cell will try to divide, and usually there is a mitotic death. There are many non-cycling lymphocytes, thymocytes, hematopoietic cells. They actually undergo into an interphase cell death. They do not progress further. They kind of get dormant. So, final and foremost, we have come upon the fact that the target is the DNA. Now, how the cell death can occur? The cell death can occur in two ways: by cell necrosis or by apoptosis. Cell necrosis we know is degenerative. Usually, that's how cell death happens. While apoptosis is programmed, it's a physiological process, and there are genetically controlled steps. In cell necrosis, there will be collapse of the cellular metabolism, there is depletion of ATP storage, the final events being there is membrane rupture, loss of lysosomal enzymes, degradation of nuclear chromatin, karyolysis, and there is always an inflammatory response to cell necrosis. While for apoptosis, there is chromatin condensation, segmentation, fragmentation of the nucleus into apoptotic bodies. There is cell shrinkage, loss of the cellular contact. Basically, the cell kills itself, and it culminates by engulfment of the cell by macrophages. There is no inflammatory response, and it can also occur very spontaneously in solid tumors. So, apoptosis is basically there is blebbing, and the cell breaks apart, and the phagocytosis takes place. While in necrosis, there is swelling of the cell, and the cell bursts out. There is nuclear lysis, and there is inflammation. Usually, apoptosis is the way to go for tumors, but in certain places, you can see necrosis also happening. There are two terms that I would like you to know: radiosensitivity and radiocurability. Radiocurability is the aggregation of the tumor at the primary or regional site, while radiosensitivity is measure of the tumor radiation response, the degree and speed of regression during and immediately after radiotherapy. There is no correlation between the two. These are just terminologies that you should know. Now, this is a very important table. This is basically how we achieve radiocurability. When we give a single fraction of 2 gray, we know all cells are not going to die. But the probability of survival after one fraction is like 50%. Half the cells will die. So half the cells survive. Then we give a further 2 gray, a cumulative dose of 4 gray. We still know that only half the cells will survive. So half the cells would be half of the surviving after the first fraction. Similarly, after 2 gray, another half will survive. After 2 gray, the rest of the half will survive. So you can see it's not linear, it's exponential. So we basically want the probability of survival to remaining cells to be less than 1. And that is usually achieved for squamous cell carcinoma after 62 or 64 gray. So the whole process of giving fractionated radiotherapy to a certain dose is basically based on this table. That we know that the gross tumor is 10 to the power 10 number of cells. And we know that gross tumor to be reduced to less than 1 cell will need more than 60 gray. So this is how we basically came across the empiricism that we are working. This is how how radiobiology actually contributed to the decision at what is the basic dose for a gross tumor. Radiosensitivity can be influenced by certain factors like hypoxia. The less the amount of oxygen, the less the fixation of radiation damage, the radiation damage that we had discussed earlier. Now, the amount of clonogenic cells. See, a tumor is more radiosensitive if it has more proliferating cells. But then there is also an issue because these after exposure to a certain amount of radiation, these clonogenic cells undergo an adaptive process and start populating more. It's called accelerated repopulation. We'll be discussing this further. Now you can see here that the cells which are away from the blood vessel are hypoxic, but they are viable cells. So during the initial radiation, the initial fraction of radiation, the aerobic cells would be killed because of the fixation of the radiation damage. And then these further hypoxic cells would be oxygenated, and then they would become radiosensitive. Other factors influencing radiosensitivity are the inherent radiosensitivity. Some cells have better cell survival, then repair of the radiation damage, the sublethal damage, and the potentially lethal damage. Now, you can see here that if you look at X-rays and if you look at carbon ions, okay, and these are various different cell lines, how their survival curve differs. For for X-rays, how different cell lines behave differently, while for carbon ions, how different cell lines behave differently. So cell survival doesn't only depend upon the cell's intrinsic radiosensitivity, it also depends upon what irradiation are we using, how much hypoxic the tumor is. A lot of things are at interplay. So what would be a probability of tumor control? Strandquist was the first person who had actually talked about it. These are the curves that he had uh, plotted. He saw that at around 1000 rad at the time, that was what was being used as the dose, erythema appeared in skin. If only single treatment was given after 1 day, after 7 days, after 15, 20, 30, 40 days, this is how erythema increased and the dose increased. When did dry desquamation come in, moist desquamation come in, cure of skin cancer came in, and skin necrosis came in. So it was basically with these plots the initial dose was decided for treatment of skin cancers, that at what dose, how much of erythema can we expect, and in how many days should we treat the patient. So these plots were defined by von Essen, and he demonstrated that for 97% tumor control and 3% skin necrosis could be possible if appropriate fractionation was used. And we had already discussed that tumor control is a probabilistic event. So various levels of irradiation will yield different probabilities of tumor control, right? Now, if you look at this place, this side we have logarithmic cell kill in the tumor. This side we have overall treatment time. And this is if we give 30 fractions at 2 gray per fraction, then how tumors with different cell tumor doubling time would perform. If the tumor doubling time is just 2 days, they'll do better. But if it's infinite, that is very slowly dividing cell or a very slowly dividing tissue, then the tumor's survival would be better. So see, you can see that there are a lot of factors on which your tumor survival is depending and your tumor control probability is depending. Then the effect on normal tissue. It depends upon the total dose, the fractionation schedule, what's the daily dose, what's the overall treatment time, how much of volume has been treated. So for normal tissue, the radiation dose necessary to produce a particular sequelae increases as the irradiated fraction of volume of organ increases. Okay. For late radiation effects, we can think of the organ being made up as multiple functional subunits. These subunits can be reached serially or parallelly. Okay, so the injury to these normal tissues is usually caused by effect on the microvasculature or the stroma or the parenchymal cells. And what can be the final sequelae? They can be structural alterations without anatomical or functional impairment, or there can be substantial injuries like tissue destruction, severe dysfunction, even death. Now, what we talked about as functional subunits, they can be arranged serially, like for the spinal cord. If one functional subunit gets injured, the other functional subunits basically become inactive and the whole organ becomes impaired. But like for lung, where the functional subunits, or like for kidney, where the functional subunits are parallelly arranged, some functional subunits might become sterile or basically get knocked out by radiation, but other would remain active and the whole organ as such would remain functional. Now we talked about tumor control probability, we talked about normal tissue complication probability, the TCP and the NTCP. So they both basically give us a therapeutic ratio or a therapeutic window where our doses must lie. And a therapeutic gain factor can be calculated to see how efficient a particular therapy is. It is basically percentage of tumor control with therapy A versus B divided by percentage of complications with therapy A versus B. So if you look at this graph here, okay, you have the tumor control probability one side and you have the normal tissue complication probability on the other side. This is for the tumor and this is for the normal tissue. If you look at dose A, we have no tumor complication, no normal tissue complication here. But our probability of tumor control is also very poor. If you look at dose B, the complication percentage for normal tissues is less, while tumor control probability is quite high. It's not 100%, it's quite high. But if we are aiming for 100% chance of tumor control, the probability of complication also gets higher, that we do not want. So the optimal dose would come around the dose B. This is how the whole treatment, radiotherapy treatment basically works on. Getting the correct therapeutic window, the correct tumor control probability versus normal tissue complication probability. Now, if we look at this other graph, we can see that the window here is very narrow. But still, we are looking at a dose where we can get a better tumor control versus normal tissue complication. Now, if we look at the fact that we have put in chemotherapy here. Okay. So this was the graph of tumor control when there was no chemotherapy given. When there was chemotherapy given, the graph shifted to this side. At lower doses, we were getting better control. But then similar thing happened for normal tissue as well. When we were not giving any chemo, the complication rate for normal tissue was here. But with chemo, we were getting more normal tissue complication as well. Over here, you can see it. So basically, chemotherapy itself also has certain toxicity. What we want is where we would be able to get a better therapeutic ratio. If we'll be able to get a better therapeutic ratio with chemotherapy, we'll actually try to put the patient on it. While without it, then obviously we'll try to do it without it. Similarly, the intrinsic radiosensitivity that we talked about. For a radiosensitive tumor, the tumor control probability curve would always lie to the left side, while for a radioresistant, it would lie to the right side of the tissue damage, because that's how the tumor biology is. So, is there any effect of local tumor control on survival? Well, there actually is. We give chemotherapy, immunotherapy, targeted therapies, all for systemic control, but there is effect of local regional tumor control on patient survival. And in clinical experience, it has been seen with breast, prostate, lung. Now, we are coming to dose time factors. Dose time factors are basically complex interdependence of total dose, time, and number of fractions. Introduction of a particular biological effect within a given tissue volume. These are basically the things that a radiation oncologist tries to exploit. to get the best tumor control probability and the least normal tissue complication. So this is very closely related to the 4 R's of radiobiology. These 4 R's are like the 4 pillars. There is a fifth, sixth, seventh R as well, but these 4 are the main basic pillars on which the whole of radiation oncology stands. First is the repair of sublethal and potentially lethal damage. Second is repopulation of cells, which occurs between fractions. Then there is redistribution of cells. What exactly happens is, in a tumor, all cells are in different phases of the cell cycle. When we irradiate the cell, all the cells in the radiosensitive phase die. The radio-resistant ones are left, and then they move in their own cell cycle. So there is basically a redistribution of the cells. Then reoxygenation, which occurs during repeated radiation exposures. We all know oxygen is so important for radiation response. And we had seen that once the oxygenated cells die, the hypoxic cells get oxygenated. So this is the reoxygenation. Then optimal overall time also depends primarily on the doubling time of the tumor cells, the intrinsic radiosensitivity, and the alpha, which we'll discuss further. Now, we do a dose fractionation to exploit those 4 R's. Advantages include reduction of the number of hypoxic cells. There will be reoxygenation happening. There's a reduction in the absolute number of clonogenic tumor cells. The blood vessels compressed by the cancer would get decompressed, leading to better oxygenation. Then fractionation also exploits a very important thing, which is the difference in the recovery rate of normal, acute, late reacting tissues and the tumor. There is redistribution of cells within the cell cycle, and then sensitization also happens. The acute normal tissue toxicity of single radiation dose can be decreased with fractionation. So these are the different types of fractionation that we can see. Conventional fractionation is 2 gray per day. Hyperfractionation is the specific dose plus a smaller amount of dose. Like here we can see 1.15 with 2 gray, or 1.15 given twice a day. Then accelerated fractionation we can have. We can have modified acceleration fractionation. We can have split course, where we treat for some time, give a break, then again treat for some time, then give a longer break. There can be hypofractionation, where we give higher dose per day, like up to 5 gray per day. So we'll discuss all of them in detail, not in this video possibly. Out of empiricism and convenience, we have decided that the standard fractionation is going to be 5 fractions weekly, 1.8 to 2 gray per fraction. This is the conventional fractionation. Anything other than this is altered. And you have seen that there is a very narrow window between the tumor control and normal tissue toxicities. So any altered fractionation is sure to be taken with a pinch of salt. Read about it and only then apply. We basically want to shorten overall treatment time. That's why we have gone into accelerated fractionation systems. Then when you have larger than standard size fractionation, or you can give more than 5 fractions per week. So a lot of permutation and combinations have happened, out of which hyperfractionation and hypofractionation have come out to be the most common 2 forms of altered fractionation. Aim of hyperfractionation is to achieve the same incidence of late effects on normal tissue while increasing probability of tumor control. Withers and Taylor first described accelerated repopulation of tumor cells during fractionated radiation therapy. They said that it occurs preferentially after 4th week, but the molecular process basically starts after the first radiation exposure, or in patients where chemo has been given. It starts after giving chemotherapy. Neoadjuvant chemotherapy can lead to increased proliferation. So if you look at these patients where treatment gap has been given, this is basically data of 533 patients. A treatment gap has been given. There has been a treatment gap from a range of 3 days to 20 days. And we can see that the more late the treatment gap was, the poorer was the tumor control because of the accelerated repopulation. Now coming to linear quadratic equation, the alpha beta ratio that we have been mentioning everywhere, alpha is there, alpha is there. What exactly happened was, when initially people were studying the effect of irradiation on cells, they were basically trying to put it into some mathematical formula. So a lot of models had come up. There is the multi-hit target model, multi-target model. Then this is the model that has the linear quadratic equation. The model has been considered as standard currently, and is maximally dependent upon by all clinicians. They basically plotted dose versus the surviving cells. So as the dose increased, the surviving cells decreased. And they showed that there are two components of cell killing, the alpha cell killing and the one with the curvature, with the slope or the shoulder, the beta cell killing. So this gave us the equation that the natural log of the survival was alpha d plus beta d square, where d is the dose and alpha is the alpha cell killing, beta is the beta cell killing. Okay. So alpha represented the linear component, beta represented the quadratic component. Now beta was repairable. It basically, the repair was happening. Alpha is the cell kill, lethal damage. Beta is the sublethal damage or the potentially lethal damage. Basically the sublethal damage which can be repaired. That's why there's the curvature, there's the shoulder. The dose at which the two components are equal is the alpha beta ratio, and it's a measure of how soon the survival curve will begin to bend over, or that shoulder will begin to appear. So we can see this is the non-repairable process, the repairable injury. Similarly, acute tumors, acute reacting cells are similar to tumors, while late reacting cells are different. So Steele and Peacock had studied 17 tumor lines, and they said that for a surviving fraction at 2 Cray, 0.44 is from alpha component and 0.88 is from the beta component. The beta effect at the dose levels appear to be similar in radiosensitive and radioresistant tumors. Beta effect makes a very small contribution to overall radiosensitivity. It is the alpha which is it defines how radiosensitive a tumor is going to be. In more radiocurable tumors, cell killing by alpha component represents the prominent fraction of tumor cell killing. So the shape of the overall survival curve with photon differs from acutely to slowly responding normal tissues. With neutrons, this is not the shape. This is for photons. As we can see here. For neutrons, there is no difference. Acute like acting, late reacting, they all will act the same. While for photons, late reacting tissues have a survival curve as such as the yellow curve, while the purple is for acutely reacting or even tumors. Now, how do we decide upon alpha? What is alpha and what is beta? So what we do is we plot the dose per fraction against the surviving fraction, and we get a straight line. The intercept is the alpha, while the slope gives us the beta. So the ratio of alpha and beta, that is the alpha beta ratio, which we have gotten experimentally and clinically, is around 10 for skin and subcutaneous tissue. For jejunum, it's 10 to 8, we can see here. So these are basically early reacting tissues. So the alpha beta ratio is high, while for late reacting tissues, it is low, like for spinal cord 3.3, lung 4, bladder 3. Now, Withers et al. Actually proposed a method for using these survival curves. Now, what actually happens during radiation oncology practice is that we start treating the patient at a certain fractionation, then there are times when we have to change that fractionation. From 2 gray, we might need to go to 4 gray. The patient might not be able to tolerate, or our intent of treatment might change. So if or the patient had taken treatment at some other center, now they're here, a lot of things can happen. So for such clinical scenarios, they had proposed a concept of biologically equivalent dose. What exactly that means is that if we are treating a patient at 2 gray per fraction, or we are treating the patient at 4 gray per fraction, what would be the biologically equivalent dose, so that we can decide that the dose received by the patient is the same. Okay, so we know that the natural log of survival was alpha d plus beta d square. That was for a single fraction. N is the total number of fractions, so we multiply it with the n. We take the d common, and then we come upon the fact that biologically equivalent dose is the log of the survival divided by alpha. The radiosensitivity of the cell is dependent on alpha. So once we replace this log of S with the factor that we know, and with further calculation, we come upon this formula of ND B into 1 plus D upon alpha by beta. And this is basically used most commonly for calculations. But here we are accounting only for the effect of repair of cell injury. We are not talking about any other factor. There were a lot of factors which were actually influencing the effect of radiation on the tumor cell, but we are accounting only for repair of cellular injury. This is a very crude and a method of approximation, but it helps us a lot clinically. So this was the formula that we came upon finally, and we'll consider it with an example. Suppose a patient was being treated 50 gray in 25 fractions, and it was giving a certain biological effect. Now we assume that the subcutaneous tissue is the limiting parameter, and we want to treat that patient with 4 gray per fraction. So for late fibrosis, the alpha by beta would be 2. So we take the formula BED ND into 1 plus D upon alpha by beta. We put in N, which is 25, 2 gray per fraction. 50 gray in 25 fraction would be 2 gray per fraction. D is dose per fraction. Alpha by beta would be 2 here, because we are talking about late fibrosis. We are not talking about tumor effect here. We can actually apply this formula for normal tissue toxicity as well. So we are talking about the subcutaneous tissue. After calculation, we come upon the fact that the BD for subcutaneous tissue would be 100 gray. Now we were going to treat the patient by 4 gray per fraction. So what dose would amount to this BD of 100 gray? We now know the BD. We apply the same formula, right? We know the BD. We do not know the number of fractions. We know the dose per fraction would be 4 gray. So we apply it in the formula. We do a further calculation, and the total number of fractions come out to be 8.3. Now, obviously, it cannot be 8.3. It will either be 8 or be 9. So, we stay on the safer side. We treat the patient to 32 gray in 8 fractions. So, this is a very crude example, but such clinical scenarios we do come across during our practice. So, this becomes a very handy and a very simple formula to help us calculate. Now, there is another concept that we had talked about, which was the accelerated repopulation. So, if we want to account for repopulation, then we'll talk about this. N is equal to N naught to the power of E lambda T. N being the number of clonogens at time T, while N naught being the initial number of clonogens. See, the tumor started with N naught. Now it's repopulating. So, it is going to become N. So what does this repopulation depends on? It depends upon lambda and T. Lambda is the factor by which the patient would be repopulating, and lambda in itself is log E square upon T pot. That is the lambda is the speed of cell population. T pot is the effective doubling time of cells in the tumor, basically. So repopulation is basically dependent on the doubling time. So we basically get a constant here. 0.693 is log E square upon teapot. We have to subtract it from the BED equation. Now, for example, a patient was being treated to 70 gray. So the BED was coming out to be 84 gray. Okay. But the patient is repopulating as well, right? So we have to subtract that. So the effective dose actually comes out to be 63 gray only. Okay, so this is how basically repopulation affects. But this kind of makes things a little complicated, so we use it less. But we have to keep in mind that tumor repopulation is a very important factor. So, in the LQ model, redistribution of cell cycle and reoxygenation are modeled by a single term called resensitization. The immediately after a dose of radiation, the average radiosensitivity of the cell population falls and then it gradually returns. In contrast to tumor proliferation, resensitization probably increases the overall treatment time as probably increases as the overall treatment time increases. Now, we do not know much about resensitization's clinical importance to put it actually into the LQ formula. So this is where basically the limitation of the LQ formula comes, but it's very handy in clinics, so we use it anyways. Now, for brachytherapy and the radiobiological dose rate effect. Brachytherapy can be divided into three, divided according to three ways. First is by the physical placement of the source. It can be interstitial, needles are put in. Intercavitary, the source is put into a cavity. Mold, it is basically put over a surface for superficial tumors. And intraluminal, where it is put into a lumen like esophagus or trachea. Now, by duration of treatment, they can divide it into permanent or temporary. Permanent implants are like these seeds which are put into the prostate. These are gold seeds which are put into the prostate. And temporary were those interstitial or intracavitary implants. Then we can divide it by rate, which can be low dose rate, high dose rate, and pulse dose rate. Now, division by rate is what is we would want to see in radiobiology. Low dose rate brachytherapy is basically administered over a period of days, while high dose rate in a few minutes. Low dose rate is usually given by cesium-137, while high dose rate by iridium-192. Intermediate or pulse dose rate brachytherapy is basically fractionated high dose rate only. You can give it several times a day. Now, low dose rate brachytherapy can be considered as an infinite number of small fractions and there is no interfraction interval. There is basically continuous exposure of irradiation. Fundamental series of effects on low dose rate on cell death is there is lethal, irreversible, and irreparable damage. There is potentially lethal damage, whose injurious effects can be exaggerated by post-irradiation environment, and sublethal damage, which can be repaired. Heavy particle beams are alternative particle beams, which have a better dose distribution or more skin sparing than X-rays, or better stopping characteristics, or less side scatter, or they are more toxic to hypoxic cells or radio resistant cells, a lower OER or a higher RBE. But the only issue with these is that these are difficult to produce. They're very expensive and they are difficult to control. They include protons, neutrons, pions, helium, carbon 6, oxygenate, neon 10, and argon 18. The biggest benefit of them is Bragg peak. Basically, because of now we come to the boron neutron capture theory. The boron neutron capture therapy basically comes out of this formula that when boron is irradiated by a neutron, a slow or a thermal neutron, we get lithium and helium, which and a photon. So we basically want to target or label the tumor cell with boron containing compound. Then we want to irradiate it with slow thermal neutrons. The compound will capture the neutron, will go into an excited state. There'll be fission, release of energy, and these heavy ions would be driven only into the cell, not for a very long distance, into the cell. So, and they can cause then DNA damage. So, this actually looks like a magic bullet, but it's a highly localized It's a highly localized idea that we can kill a tumor cell containing boron while we can spare the adjacent normal cell. So the idea was to specifically label these tumor cells. And in 1936, this person, Locher, had actually published a theoretical account about the possibilities. And in 1950, William H. Sweet et al. Had actually started doing it for brain. They had started working with a 20 megawatt nuclear reactor. That was the major issue. First was that the inadequate tumor specificity of boron compounds to localize in the tumor. The boron compound should not localize in the tumor. There'll be poor penetration of the tissue to the thermal neutrons. The beams are difficult to produce, and there are a lot of contamination with gamma rays and fast neutrons that can lead to normal tissue damage. Thermal neutrons can interact in normal tissue and cause normal tissue damage. There are no suitable boron carrier agents. And the biggest issue is production of these neutrons. It can be in a nuclear reactor or by Californicum 252 or by particle accelerator. And we have so less amount of Californicum 252 that it is not feasible. Particle accelerator we would need of the level of a hadron collider. So that these are major reasons why there has been a lack of progress in BNCT. I know today's video was very long. I am extremely sorry. I'll try to keep it as short as possible. But please do comment. And thank you so much for your time.